Welcome everybody to a Striatech hosted journal club. My name is Thomas Münch and I'm one of the founders of Striatech. In our journal club series, different scientists present their projects and new data, highlighting the use and the application of our OptoDrum device. Today I'm very happy and pleased to welcome Janos Groh as our speaker. Janos works at the University of Würzburg in Germany in the lab of Professor Rudolf Martini. He will talk about assessing neuroinflammation-related damage by monitoring the retinotectal system. Before Janos' talk, I will give a short introduction. Please let me highlight that throughout this event, you can ask questions and we will have time to answer and discuss your questions at the end. A quick word about Striatech. Striatech is a dynamic biotech company from Tübingen in Germany. We specialize in the development of neuroscience testing tools, in particular for vision research. The Striatech founders are all experienced neuroscientists in the field of vision research. Today's talk is not really about vision, though. The research focus of Janos Groh lies in neurological disorders. Many such disorders of the nervous system are rare diseases that are genetically caused. Their specific underlying mutations can be very diverse, and so can be the cellular and the subcellular structures that are affected by these mutations. What is listed here are a couple of examples that Janos will mention in his talk. Generally, these diseases are very poorly treatable, and because of the diversity and the rarity of the individual disease, it is difficult to find specific treatments. Over many years of research, though, Janos has found that there is a common theme to many of these diseases. In some way or another, these diseases activate the immune system, and via neuroinflammatory processes, the immune system then turns against the body's own cells. Some of the devastating consequences of these diseases then stem from these secondary effects, from the destruction of glia and neurons by the immune system. Because of these common secondary effects, targeting the immune system with immunomodulatory drugs might offer therapeutic opportunities for several diseases that are currently not treatable. Janos will tell us about an example where preclinical results have led to the first treatment approaches in a fatal lysosomal storage disease affecting children. What is more, Janos has shown that not only diseases, but also the normal aging process triggers the same type of neuroinflammatory effects. Neural decline in old age may therefore also be counteracted with immunomodulatory treatments. As Janos will show, myelin and myelinated axons are among the primary targets of these neuroinflammatory processes. The optic nerve is such a target structure, and this is how we come back to the visual system. As it turns out, the retinotectal system, so the retina, the optic nerve, and the tectum, are very convenient to monitor disease progression and the effects of treatment. And at all levels, at the subcellular and cellular level, at the tissue level, at the physiological level, and also at the behavioral level. This last point, the evaluation of visual abilities at the behavioral level, is where Striatech's OptoDrum can help, as we will see in Janos' talk. The OptoDrum takes advantage of the Optomotor Reflex. The Optomotor Reflex is a visual reflex that makes the animal follow global motion in the environment. Experimentally, you can evoke the optomotor reflex with the rotating stripe pattern surrounding the animal as seen here. Janos could observe with our OptoDrum how vision became worse in his animal models and how treatment, the modulation of immune responses, could prevent this functional decline. With this, I'm very happy to introduce Janos Groh. Janos studied biology with a focus in neurobiology and genetics at the University of Würzburg. He got his PhD in the laboratory of Professor Rudolf Martini at the Department of Neurology, where he developed a strong interest in the complex interactions of the nervous system and the immune system, and the contribution of neuroinflammation to various genetically mediated neurological diseases. He continued his work on these topics as a postdoctoral researcher and project leader, and recently broadened his research scope to include aging-related degenerative processes in the nervous system. Before you start, Janos, let me remind the audience that you will be able to ask questions throughout the event, and also give a thumbs up to other questions. A quick word about asking questions. The interface can be a bit confusing. When you want to ask a question, the cursor by default is in the name field. Giving your name is optional, but you have to click into the question field before you start typing your question. The Q&A panel on the right will stay available for a couple of days after this live event. So if you are currently watching a recording of the event and Q&A is still available, please make sure to enter your question and your name so that we can back to you with the answer. And we are very much looking forward to your questions. So please, Janos, go ahead and with your presentation.
Thank you, Thomas. So as introduced, I will talk about neuroinflammation-related damage in the retinotectal system. As you can see in this outline, I will first introduce our working concept exemplified by specific disease models that carry glial gene defects. In the second part, I will describe how we take advantage of the properties of the retinotectal system to assess damage and also therapeutic effects in disease models that carry glial or also lysosomal gene defects. And finally, I will describe how similar mechanisms are relevant for the normal aging process in mice without specific gene defects. So the main interests of our group focus on models of neurological diseases that are caused by primary perturbation of the nervous system, for example, by specific gene defects. But these diseases are also often accompanied by neuroinflammation. And such secondary immune reactions can act as a strong modifier of the disease outcome, and therefore we postulate that immunomodulation might offer therapeutic opportunities for several of such diseases that are typically poorly treatable and often rare orphan diseases. And initially, this concept was developed from studies in the peripheral nervous system. As mentioned, I was lucky to start working in Rudolf Martini's lab on one of several models of Charcot-Marie Tooth disease type 1, which is an inherited peripheral neuropathy. And there we found that Schwann cells, the myelin-forming cells in the peripheral nerves that carry specific myelin gene defects, and endoneurial fibroblasts communicate with macrophages, so the innate immune cells in the nerve. And this interaction induces a chronic nerve inflammation, which is important to drive myelin pathology and also clinically relevant axonal damage. An important pathway for macrophage activation in these disease models was colony stimulating factor 1 or CSF1 signaling. And this concept was also tested in the central nervous system. Here the Martini laboratory could really concentrate in a series of studies on a mouse model of Pelizaeus-Merzbacher disease, so an inherited leukodystrophy that overexpresses the proteolipid protein, which is the major myelin protein in oligodendrocytes of the central nervous system. This model was initially generated in Klaus Navis' laboratory, and the group could demonstrate that activation of immune cells contributes to the prominent demyelinating phenotype and also to the milder axonal damage observed in this mouse model. So we tried to test the relevance of these observations for more frequent chronic inflammatory diseases of the central nervous system. And in the literature, we found 2 case reports of patients with a diagnosis of progressive multiple sclerosis that describe point mutations in this proteolipid protein 1 gene, which usually results in PMD or also spastic paraplegia type 2. So in collaboration with Alan Peterson's lab from Montreal, we generated new mouse models that carry exactly these described point mutations, PLP mutant mice. And these novel models indeed reflect some important features of progressive multiple sclerosis, but also SPG type 2. And they show myelin defects and also mild demyelinating phenotype. However, again, clinically much more important is that they develop prominent axonal damage, as you can see by this hallmark sign of axonal spheroid formation in the white matter. The myelinated axon is swollen, it is filled with organelles and dense bodies, and ultimately this damage results in neurodegeneration. It results in brain atrophy and also in impairment of sensory and motor function in these genetic disease models. Supporting our concept, we also observed chronic inflammation in the white matter. There was microglial activation, as seen on top, with a labeling of CD11b, and also an infiltration of mostly CD8 positive cytotoxic T lymphocytes in these PLP mutants, detectable if you compare them to wild-type mice. And such CD8 positive T cells normally recognize and fight infected or malignant cells. However, in case of autoimmune diseases like multiple sclerosis, they can also do unwanted damage in the nervous system. To test the impact of these adaptive immune reactions, we used crossbreeding approaches with distinct knockout mice to generate myelin mutants with deficient or aggravated immune reactions. Eliminating mature adaptive immune cells with RAC1 deficiency caused an attenuation of the disease severity, and in contrast, hyperreactive adaptive immune reactions by deficiency for programmed F1, PD1, which is a regulatory checkpoint molecule on T cells, aggravated the disease severity in these PLP mutant mice. And for such experiments, we routinely analyze the retinotectal system in mice. And this system comprises distinct, well-characterized and also accessible compartments of the central nervous system, and it allows both invasive and also non-invasive monitoring of disease progression. So focusing on retinal ganglion cells, we can assess pathological alterations to the cell body and dendrites in the inner layers of the retina. We can analyze a white matter compartment with long axons myelinated by oligodendrocytes in the optic nerve, and we can look at synaptic terminals in the superior colliculi of our mouse models, our disease models. To study early alterations of axonal integrity in PLP mutant mice, we performed stereotactic injections of a fluorescent tracer into the superior colliculi, which is then retrogradely transported via the optic nerve to the retina. Analyzing the tracer accumulation in retinal flat mount preparations revealed that actually one of the earliest changes we see in PLP mutant mice is an impairment of fast axonal transport. So there's a delayed accumulation of the tracer in PLP mutant mice before there is detectable neuron loss. And this impairment is mediated by adaptive immune cells, since there is less impairment of transport in RAC1 deficient PLP mutants. The tracer accumulates normally in the retina. Subsequent analysis showed that this early reduction or this focal block of axonal transport after tracer injection seems to really occur focally in axonal spheroids in the optic nerve. So the immune cells accumulate at specific axonal domains of the nodes of Ranvier in these disease models. And exactly at these juxtaperonodal domains seen here, we see an accumulation of organelles and dense bodies in these myelinated axons. And these accumulations progress to form axonal spheroids, which increase in size in these older mutants, and which we can label with SMI32 antibodies that recognize non-phosphorylated neurofilaments. The axons ultimately become transected, resulting in loss of retinal ganglion cells, here labeled with the specific marker RBPMS in retinal flat mount preparations. And all of these sequential changes are much more pronounced in PLP mutants than in REC1 deficient PLP mutants, which lack mature adaptive immune cells. So adaptive immune cells contribute to axonal damage and neurodegeneration in these PLP mutant mice. We also established non-invasive readout measures to track this progression longitudinally in form of optical coherence tomography or OCT in living mice. So the mice are anesthetized and we can make high-resolution scans of individual retinal layers. By measuring the thickness of the innermost retinal composite layer, which comprises the nerve fiber layer, the ganglion cell layer, and the interplexiform layer, we can follow progression of retinal atrophy, which correlates with neurodegeneration in PLP mutant mice. And again, REC1 deficient PLP mutants, here in green, show a strong attenuation of retinal atrophy, reflecting that adaptive immune cells contribute to retinal thinning and neural damage. So in conclusion, we also saw in these novel genetic models that are relevant, in our opinion, for progressive forms of MS or SPG2 with a primary myelin gene defect in oligodendrocytes, that secondary inflammation comprising these CD8 positive T cells is really an important driver of axonal damage, contributing also to the clinical phenotype. And we therefore tested translational pharmacological approaches with well-established immunomodulatory drugs, fingolimod and teriflunomide, and the efficacy of these drugs in these progressive disease types is still relatively unclear. And indeed, when we use the right treatment time point and also the right drugs, we could attenuate inflammation, we saw attenuated neurodegeneration and also less clinical disease. And demonstrating really the suitability of our translational longitudinal readout measures like OCT, therapeutic efficacy was reflected by a reduced thinning of the retina upon treatment with both of the drugs here shown at different time points. In addition, we also performed genetic and also pharmacological experiments to target the innate immune reactions that we observed in PLP mutant mice. To do this, we knocked out the pro-inflammatory cell surface molecule sialadhesin, or SN, cyclic 1, on myeloid cells in PLP mutants. And in addition, we performed pharmacological depletion experiments of these pro-inflammatory microglia using PLX3397, an inhibitor of the receptor for this important myeloid cytokine CSF1 that I already mentioned. And both of these approaches modulated the CD8 positive T cell reactions in the central nervous system and resulted in attenuation of the disease. And this reflects a cooperation between CNS resident innate immune cells and the observed adaptive immune reactions in PLP mutant mice. To further investigate the contribution of neuroinflammation to functional decline, we performed measurements with the OptoDrum device, which allows unbiased assessment of visual acuity. As you can see, there's a progressive decline in visual acuity, nicely correlating with the degeneration of retinal ganglion cell axons in PLP mutants. And again, this decline is significantly attenuated by sialadhesin deficiency up to 18 months of age, demonstrating that neuroinflammation is involved in both structural and also functional decline in this disease model. So in summary, we could show that primary gene defects that affect myelinating glial cells of the peripheral, but also of the central nervous system, can result in secondary neuroinflammation that strongly modifies disease outcome. However, we were able to show that this concept is not restricted to gene defects in myelinating glia, but is also valid for models of rare but fatal lysosomal storage diseases. Neuronal ceroid lipofuscinosis, or CLN diseases, are really horrible neurodegenerative diseases that affect children. and they are caused by lysosomal gene defects and result in blindness, mental decline, epileptic seizures, and ultimately premature death. And we have shown in 2 different models of infantile and juvenile CLN disease that, again, secondary inflammation modifies disease outcome. Also in these models, we use the retinotectal system as a surrogate for CNS pathology. And interestingly, as observed in many neurodegenerative diseases and also in our other disease models, axonopathic alterations really present early before neuron loss also in these CLN diseases. So there's a progressive accumulation of organelles. There's again this formation of axonal spheroids that starts at the juxtaperonodal domains, as you can see here. And as mentioned, these axonal spheroids, they can be labeled with antibodies against non-phosphorylated neurofilaments, and they also show activation of specific pro-degenerative molecules. Moreover, we again observed an increased number of mostly CD8 positive cytotoxic T cells during disease progression in CLN disease models, and we also found that sometimes these CD8 positive T cells were found in association with damaged axons. Using again crossbreeding approaches, we could then demonstrate that CD8 positive T cells and their cooperation with activated microglial cells or myeloid cells plays an important role in the neurodegenerative disease process of these CLN models. So again, crossbreeding with REC1 deficient mice, or also with mice that lack this pro-inflammatory myeloid cell surface molecule, CL adhesin, caused an attenuation of axon degeneration and neuron loss, and also an amelioration of the clinical phenotype. Similarly, pharmacological depletion, again using the CSF1 receptor inhibitor PLX3397 to deplete pro-inflammatory microglia, modulated CD8 positive T cell reactions in the CNS and resulted also in attenuation of the disease. Using translationally relevant readouts like OCT and also OptoDrum, also here demonstrated a reduction of retinal thinning and also attenuated decline in visual acuity upon depletion of detrimental microglia. Following our concept to repurpose established immunomodulatory drugs for rare diseases, we could then show that fingolimod and teriflunomide, so these immunomodulatory drugs, the treatment with these drugs is also able to dampen inflammation and neurodegeneration in CLN disease models. Again, we could demonstrate this in the retinotectal system, but we also validated that this really reflects more general disease also in other brain regions. And since CD8 positive T cells were then also detectable in human brain autopsies of CLN disease patients, this now resulted in a first small off-label therapy for CLN disease using fingolimod in few patients, which is really exciting for us. So one question that is often relevant for us is why we are interested in secondary processes in such rare genetically caused diseases. We always felt that it is very important to study such orphan diseases and also that they offer great opportunities to learn for other more frequent pathological conditions. And considering the commonalities of the studied disease models, with some known aging-related pathological alterations in the central nervous system, we now focused on similar mechanisms in the normal aging process. So in the third part, I'm happy to tell you about these studies that were published last month in Nature Aging. And indeed, we could observe that in optic nerves of normal aging mice without disease, there are alterations in myelin integrity, and there are also signs of axonal damage, again represented by the formation of axonal spheroids. And these spheroids, when we label them with our antibody against non-phosphorylated neurofilaments, are rarely detectable in 12-month-old, so adult mice, but they seem to accumulate in 24-month-old mice, which we consider as aged, as you can see here. So using optical coherence tomography at various ages from 2 up to 24 months of age, showed that actually there's not a linear increase of pathology with age, but that thinning of the inner retina here occurred only at 24 months of age. And when we quantified retinal ganglion cells in flat mount preparations, there was only a very mild loss of RBPMS positive retinal ganglion cells detectable at 24 months of age, while CHAD positive displaced amacrine cells, which do not project to the optic nerve, these cells remained unaffected. So to better explain this thinning of the inner retina, we then looked at dendritic arborization of alpha retinal ganglion cells in Thy1 YFPH transgenic mice that express YFP in a small subset of neurons. And this allows us to trace the dendritic arbor and then perform Scholl analysis. And this tracing demonstrated that there is reduced dendritic length and also reduced dendritic ramification in aged retinal ganglion cells compared with adult retinal ganglion cells. And this is a typical reaction of retinal ganglion cells after axonal damage. Indeed, in the optic nerves of these YFP transgenic mice, axonal spheroids and again also discontinuous transected axons could be observed. And moreover, when we looked at the superior colliculi of the mice, we also saw reduced densities of axon terminals and collaterals, which also indicates ongoing axonal degeneration. As a functional readout, we performed OptoDrum analysis of visual acuity, and we did not detect significant decline in visual acuity at 24 months of age. Only at even more advanced age, so at 28 months of age, we saw that there was significant impairment. And this might indicate compensatory mechanisms at 24 months of age when there already is axonal degeneration detectable. We again investigated neuroinflammation as a possible modifier of neural damage in the aging central nervous system. And interestingly, these SMI32 positive fibers and also irregularly shaped axons in 24-month-old, so in aged but not in 12-month-old YFP transgenic mice were occasionally contacted by CD8 positive T cells, again suggesting that there might be a detrimental impact of adaptive immune reactions. So when we quantified this, we revealed that these SMI32 immunoreactive axonal profiles also steeply increase in number between 18 and 24 months of age. And similarly, the CD8 positive T cells, they also accumulate in the optic nerves, and they follow a very similar pattern fitting to axonal spheroid formation. So importantly, when we then investigated aged RAC1 deficient mice, which of course lack adaptive immune cells, axonal spheroid formation was significantly reduced in aged mice. A detailed electron microscopic quantification of total axon numbers, a really prominent degeneration of axons was detected in these aged mice. And again, this was strongly attenuated by RAC1 deficiency. So also in aging, adaptive immune cells drive the degeneration of myelinated axons. Optical coherence tomography also demonstrated that aging-related decline of retinal thickness was attenuated by RAC1 deficiency. And similar to our disease models, we observed that inflammation and axon damage was not really confined to the retinotectal system, but it occurred also in other CNS white matter regions, such as the cerebellar white matter and also the corpus callosum. So we therefore also investigated the impact of inflammation on typical aging-related behavioral phenotypes that are independent of vision. And as you can see, RAC1 deficiency in aged mice also resulted in an improvement or less severe impairment of rotor rod performance, and it also prevented aging-related cognitive decline as measured with the novel object recognition task. We concluded that inflammation and especially these CD8 positive T cells really contribute to motor and cognitive decline in aging, by causing axonal damage and degeneration in multiple white matter compartments. In collaboration with Wolfgang Kastenmüller and Emanuel Saliba from Würzburg, we then characterized these apparently very important CD8 positive T cells in the brains of adult and aged mice. We isolated the cells by FACS and performed transcriptomic analysis using single-cell RNA sequencing. So in these projections on left, each dot represents one CD8 positive T cell, and these different colors represent different clusters or subtypes of which we obtained transcriptomic information. And our analysis revealed that these CD8 positive CNS-associated T cells are heterogeneous and that there are specific subsets of activated cells in the parenchyma that seem to specifically accumulate in aged brains. So these subsets showed expression of markers that indicate activation, chronic T cell receptor stimulation, and also cytotoxicity. For example, LAG3, here enriched in the red, cluster 1, and granzyme B, here enriched in the red and the green clusters 1 and 2. So to confirm really the role of these CD8 positive T cells and investigate the effector mechanisms and also the cognate T cell receptor dependency, we performed bone marrow transplantation into middle-aged RAG1 knockout mice and then investigated the bone marrow chimeras at 24 months of age. When we performed wild-type bone marrow transplantation, seen here, this re-established the T cell recruitment, axonal spheroid formation, and also axon degeneration in aged RAG1 knockout mice. In contrast, when we transplanted CD8 knockout bone marrow, this only restored the CD4 positive T cells, but it did not restore spheroid formation or axonal degeneration. Additional experiments using bone marrow transplantation demonstrated that really it seems to be specific subsets of these CD8 positive T cells which drive axon degeneration in a granzyme B and cognate T cell receptor dependent mechanism. So cells recognizing an ectopic antigen did not do any damage. Interestingly, this inflammatory process in the aging white matter also represents a risk factor for further insults. When we mimicked peripheral infection by intraperitoneal LPS injection, this is known to induce systemic inflammation, which can exacerbate ongoing pathology in the central nervous system. And indeed, a single injection of LPS fostered CD8 positive T cell reactions and axonal degeneration in aged but not in adult mice 4 weeks after the systemic injection, the intraperitoneal injection. And this again was also dependent on the CD8 positive T cells. So this did not occur in aged CD8 knockout mice after LPS injection, as you can see here. So LPS injection also resulted in an aggravated loss of retinal ganglion cells in aged wild-type mice, as you can see here by the quantification, but it did not result in neuron loss in adult mice, and also again it did not result in increased neurodegeneration in aged CD8 knockout mice 4 weeks after the injection. And this was also reflected in functional decline. So on the right, I think this is a very interesting experiment where we used OptoDrum to assess visual acuity. In 23-month-old mice, you might remember that we did not see a significant decline of visual acuity. However, when we analyzed the same mice 1 month after LPS injection, the exaggerated neural damage caused a decline in visual acuity. And again, this was dependent on the CD8 positive T cells. So this did not occur in aged CD8 knockout mice that we analyzed before and again also after LPS injections. So these are the same mice. So this demonstrates that neuroinflammation in the aged central nervous system is susceptible to systemic stimuli and might have important implications for older adults. So in a translational approach, we then analyzed brain autopsy material from adult and older normal human frontal white matter without disease. And we received samples from 25 to 50-year-olds and a group of 70-plus-year-olds from the Edinburgh Brain and Tissue Bank. And indeed, also here, there was a significant accumulation of mostly CD8 positive T cells, but not of CD4 positive T cells detectable. And we found increased numbers of SMI32 positive axonal spheroids. And some of these CD8 positive T cells in humans older than 70 also expressed LAG3 and granzyme B. And again, were found in proximity to SMI32 positive perturbed axons, suggesting that also here they might cause axonal damage during aging, similar to what we could show in the mice. So this is yet unpublished data, but also in aging white matter of mice. Now again, we obtained data that microglia cooperate with T cells to drive these degenerative alterations. We observed pro-inflammatory microglial activation and also accumulation of autofluorescent storage material in some of the phagocytes, and this reflected an accumulation of lysosomal material due to extensive myelin phagocytosis. Again, using single-cell RNA sequencing of sorted microglia from the brain and also of oligodendrocytes from adult and aged mice, we observed different clusters of pro-inflammatory aging-associated microglia, including subsets reflective of antigen presentation, for example, but also of myelin phagocytosis and protection against oxidative damage. So now we can perform bioinformatic analysis of these transcriptomic data, and this allows us to really look at the similarities and also the differences between normal aging and also these genetic disease models regarding these glial subsets. And finally, genetic and also pharmacological targeting of microglia in aged mice also resulted again in modulation of CD8 positive T cell reactions and axon degeneration, again arguing for the interplay between these innate and adaptive immune cells. So we think that our studies really took advantage of the compartmentalization and also the accessibility of the retinotectal system, and they allow the following conclusions. Neuroinflammation modifies structural and functional decline of the central nervous system white matter in rare genetic disorders and also during normal aging. Adaptive immune reactions, and especially those by CD8 positive CNS associated T cells, play a major and previously underestimated role in CNS inflammation. Modulating inflammation offers distinct therapeutic opportunities of high translational relevance. And we think that studying rare diseases with a clearly defined primary cause, for example, a genetic cause, can offer insights into more general mechanisms like aging that basically affect all of us. And finally, I would like to thank, of course, all of the collaborators that contributed to these studies, the funding organizations, the Martini Laboratory, and all of you for your attention. Of course, I'm happy to take questions.
Thank you, Janos, very much. That was really great. We already got quite a few questions, and if you don't mind, I'll dive right in. Let's see. So one question was whether you have any data or hint how this immune response in the central nervous system is initiated. So what causes the inflammation and what causes the accumulation of T cells?
So this is a very important question that really probably depends on which condition that we are talking about. So in our models that where the mutations affect specific cells, I think the answer is probably most straightforward. In these models that carry glial mutations, we know that based on our transcriptomic and also other data, the mutations cause activation of specific stress signaling pathways and also expression of inflammation-promoting molecules or cytokines directly by these glial cells. And we think that this is important for communication with immune cells, for example, microglia, which then directly or maybe also indirectly can initiate these adaptive immune reactions. For example, also by expressing chemoattractants or stimulatory molecules for T cells. In aging, there might be some similarities because we also see most of these inflammatory reactions in the white matter. So also there, in our transcriptomic data, we see some similar alterations, but also some differences. Regarding the lysosomal storage diseases, there it's the defects, of course, might affect multiple cell types directly. So also neurons, for example. And there, indeed, a lot of the inflammation is also going on in the gray matter. So there are clear differences also.
Okay. Do you know if there is any tissue or cell type specificity for your suppressor mutation?
The suppressor mutation, which mutation are you now?
I think suppressing the immune response.
So the REC1 deficiency?
I assume so, yeah.
Okay, so so REC1 deficient mice, they are unable to form mature T and B lymphocytes overall. So and REC1, there are some reports that it might be expressed in the central nervous system also, but our bone marrow transplantation data clearly show that the REC1 deficiency, the effect does not affect the central nervous system because we can restore the damage basically by transferring wild type T cells. So the effect is clearly immune dependent and not caused by other effects of REC1 deficiency in the central nervous system.
Okay, and another question was what the CD8 plus T cells, what they are targeting. Do they recognize a specific antigen?
So maybe, we do not know exactly which specific antigens or antigen is recognized by these CD8 positive T cells. So our data and also data from other groups indicate that to do their damage, they have to be antigen specific. There also is clonal expansion of these CD8 positive T cells. Yeah, because they mostly infiltrate the white matter and the oligodendrocytes show cell stress and upregulate, for example, MHC molecules, we speculate that they might recognize and target myelin components. So, and this might then indirectly damage the axons. Again, in the lysosomal storage disease, they might also target neuronal structures directly. So we do not know the specific antigen, but we have some ideas.
Mm-hmm. All right. Another question was, what comes first, the T cell appearance or the microglial cell activation? So
we see both things happening quite early in all of the diseases when we see the first pathological alterations to the axons, I would say. Maybe slightly earlier we see microglial activation, I would say.
Mm-hmm.
But then we quite rapidly also see these adaptive immune cells increasing in number.
Okay. You showed these images of immune cells at the nodes of Ranvier that form spherules there. Were you able to identify their cell types? Were resident immune cells present there?
So there are definitely resident immune cells present near the nodes. We see activated microglia near the nodes, near these compartments. But we also see that the CD8 positive T cells, for example, seem to accumulate specifically or at a relatively high frequency at these juxtaperonodal domains where we see the first alterations of the axons. So we see quite a good spatial correlation between the resident and also infiltrating immune cells with the formation of these spheroids.
Mm-hmm. Yes, another question was whether you used ERG, the electroretinogram, as a functional readout as well.
So we did not do ERGs. We would definitely be interested in doing them, but we didn't do it yet. No.
Okay. So one question was, you saw this at time point 24 months in your aging mice that the optomotor reflex was not affected yet, but at 28 it was. While you did see some, your other morphological hallmarks already happened at 24 months of age. Do you know what other biological things are happening between 24 and 28 months that might explain the optomotor decline in those 4 months?
So we do not have really solid data on this. We speculate that there is compensation going on at 24 months of age. So, but we do not know exactly how this is achieved. So you could imagine that you need a specific threshold of degeneration to get above kind of a readout threshold that you can detect these alterations. I think this is also pretty typical for neurodegenerative disorders, that you need a certain amount of damage that it becomes detectable in your functional readouts. Or as mentioned, there might be compensation. We do not know exactly how this compensation might look. It might be structural or also reorganization of the ion channels, for example. There are a lot of possibilities, but we do not know exactly what is the reason.
Another question was whether you know if neuroinflammation has a role in Alzheimer's disease.
Well, there's definitely a lot of literature around showing that also in Alzheimer's disease, there is neuroinflammation happening. Recently also it has been described that also in Alzheimer's disease, they find, at least in the CSF, they find CD8 positive T cells. So we could imagine that maybe some similar reactions also occur in Alzheimer's disease. But we did not work with Alzheimer's disease models, but of course there's a lot of literature also focusing on the neuroinflammatory aspects of Alzheimer's disease.
Okay. Another question is whether you know whether the blood-brain barrier is degraded in aged mice compared to younger mice.
So there are actually, I would say, different reports in the literature. Some report that there is alteration in the blood-brain barrier integrity. Other reports there's nothing. We didn't look at in detail at this. What we can say in our myelin disease models, where we see these T cells, we don't see dramatic changes in blood-brain barrier integrity. So, but we can exclude really subtle differences. So. I think the question is targeting towards the route of entry for these T cells. There are different routes possible, so we do not really know exactly how the T cells enter the aged central nervous system. And we did not really focus much on the blood-brain barrier integrity, but there are studies out there.
Okay, then there's a question that asks about tissue culture or neuronal culture with human cells, whether it would be worth to repeat some of these experiments with cultured human neuronal cultures.
So it's difficult to say. I mean, maybe some aspects you might also get in vitro, some of the aspects. For example, I don't know, treating neurons with inflammatory cytokines, but it's not our favorite experiments, and we see a lot of interactions between many different immune cell types, and also neural cell types, glial cell types, and so on. So for us, it's always more fascinating, and also we think it's really important to study the interactions between many of these different cell types, and this is really, I think, more difficult to achieve in vitro. And also you don't know really how well what you then find in vitro reflects what is going on in vivo. So we are not the biggest fans of these in vitro experiments.
Okay, here's a question that I like, that asks what immunomodulatory drugs could you imagine could be used in aging? So what is the pill that I have to take to stay young forever?
So it's difficult to say. I will not make any recommendations now. So, and I think also, yeah, it's a difficult question. So we do not want to say that we can simply suppress immune reactions and then become immortal. I think the take-home message should be more focused on trying really to improve quality of life in people that suffer from age-related neurological dysfunction or something like this. So, of course, it might also have huge implications for age-related diseases, as you mentioned, Alzheimer's disease. Yeah, but it's still unclear, I think, which immunomodulatory approaches might be feasible to do this without also too many side effects, for example. I mean, there already is immune senescence in the periphery during aging, so I think we have to be careful. But nevertheless, I think that indeed immunomodulation might offer some benefit in aging. But I can't tell you a specific compound right now.
That's unfortunate. All right, so there's another question that refers to your experiments with a rotor rod that showed that your aged mice have motor deficits. And so the question is, if you have animals that have motor deficits, whether the motor deficit as such would impair or influence the readout of the optomotor measurements, where, you know, obviously the movement of the animal in response to the visual stimulus is measured, or if this is still a truly visual phenotype that you see there.
So we think it is a visual phenotype. We have also observations in other disease models where we clearly see very early effects in the OptoDrum regarding visual acuity, and rotor rod impairment comes much later, and it doesn't really affect the optomotor reflex. I could imagine maybe that if you have a really severe motor phenotype, then maybe you might see some effects also on this optomotor reflex. But with a relatively mild phenotype we see in these aged mice regarding this motor coordination, I think we do not have an effect on visual acuity.
Mm-hmm. And do you have any idea about the role of B cells in these neuroinflammation models?
So we didn't do very specific experiments to target B cells, for example, using B cell deficient mice or something like this. But what we know is that when we, for example, do these CD8 knockout bone marrow transplantations, we have B cells, but without the CD8 cells, the B cells do not seem to do much damage. So what I can tell you is that the B cells at least need the CD8 positive T cells to do the effects that we see. Mm-hmm.
Yeah. There's a question about whether the optomotor readout is maybe not sensitive enough for these measurements. So you mentioned two examples now where motor decline is visible after you see the visual decline, and the example that you showed us in the talk is just the opposite. So do you have a take on that? On what is a good readout under what circumstances?
I really think it depends on your model and which systems basically are affected by the disease, by the pathology. And I think you have to really find this out depending on the specific model you are working with. I mean, with the aging, of course, you also have some effects in the peripheral nervous system, and you have much wider effects that affect maybe also the motor phenotype than only specifically in specific pathways of the central nervous system. In contrast, if we look at, for example, these PLPmut mice, that's really clear that it's mostly an effect in the central nervous system. So this could explain some of the discrepancies in this.
Mm-hmm. Okay, so maybe one last question before we run out of time. So it says for your MG depletion experiment, when do you think is a good time window, and how long did you treat the animals, and whether it is the depletion or the repopulation that is important for the beneficial effects?
So it depends actually also on the condition, I think. So we used lower doses and long-term depletion experiments for these microglia depletion experiments. So we do not want to get rid of all microglia and then repopulate. We actually use a constant low-dose treatment regime. So, and yeah, in our experience with these chronic progressive disease models, the earlier you start, the better it is for the outcome. So this we see with our treatment approaches, and this also seems to be true for the microglial depletion experiments.
Mm-hmm. Okay, well, Janos, thank you so much for this fantastic talk and this really, really great data. I'm very happy that you presented this here today, and also thanks to the audience for terrific questions. If you're watching this not live but a recording, please feel free to enter more questions into the Q&A as long as it is still available, and we will collect those questions and answer those as well. Also looking forward to welcoming you to the next Journal Club, which is in roughly 2 months, towards the end of July. And thank you everybody for your attention and for visiting us here online today. Goodbye.